Okay, so if we're reviewing that, so basically what it is, we're talking about this, right? The glycosidic link here. Okay, so basically what the whole concept is when you have polymers, you have monomers, which are like the building blocks of polymers. And so we're basically discussing how do we build those polymers. And so the first sort of reaction is a condensation reaction. And what happens there is you have these two monomers and you basically link them together by removing a water, so an H2O molecule. And so you can see here how you have this, which is a short polymer, and here you have an unlinked monomer. And so you have an H and you have an HO. And so dehydration, if you're dehydrated, that means you need more water. You're basically taking away water. So I'm removing this water, and when this happens, I form a bond, and so that's the dehydration synthesis. The other one is called a hydrolysis, which is the breaking down of these bonds by adding in the H2O. So here you have a bond, and I'm adding in water, so hydrolysis, and when that happens, I end up forming two different things. And so basically, in a disaccharide, we bond two monomers, or two monosaccharides, and how we bond that is by removing the HOH, which is a dehydration synthesis. I removed the water here. And when that happens, we call that a glycosidic linkage. And so you can see here we have two glucoses, which are two monomers, and we're adding them together. So if I regularly added it, I would get C12, H24, and O12. But we're losing a water, so I'm taking away one hydrogen and two oxygens, which is why, no, two hydrogens and one oxygen, which is why it goes from H24 to H22 and O12 to O11, if that made any sense. Did you get that? Or is it?
Yeah, that makes sense. Thank you.
Okay. That's that. Do you guys want to go over like the Frqs now, or do you want to just like talk through the PowerPoints?
So if no one has any questions, we can just go over the FRQs or like this PowerPoint or chapter 4.
I think Canary said let's do the FRQs. So let's do that. Okay, so I'm not completely sure about all these either, so I think it's like good for us to kind of like work through them together. So yeah. Okay, the first one, the protein that surrounds the yolk of an egg is a clear liquid when raw and white solid when cooked. So why cooking causes this change? And so I think with this one, I have the cliff notes, so the answers are in the cliff notes. So I think the big thing that they talked about with this one was, okay, well anyways, while I'm looking for it, I think the big thing that we learned about in proteins was that proteins can easily, like when you look at temperature changes and you look at pH, when you look at these different things, it can cause denaturation or it can change the protein to quickly change. And so that concept is what we're discussing here. So yeah, so we said that, and if we said that basically when a protein is heated, it begins to lose its I'm just reading the answer and then we can explain it. So it says, when a protein is heated above a critical temperature, it loses its 3D structure. When the secondary, tertiary, and quaternary structures of a protein break down, as they will when excessively heated, the structure is destroyed. So like we were saying, in a protein, one of them, I think, let me look at the slides, it's like clearly states it here.
Wait, Anna, where is this in the book?
It's in the Cliff Notes, it's on page 28.
Okay, thank you.
So it says denaturation. Shows shape change in a protein is biologically inactive, and this happens with pH, salt concentration, or temperature. So what the answer is basically saying is that when your protein is heated, so when you have a very large temperature change, the 3D structure of it is going to end up changing. And we learned that structure determines function. And so when that structure is gone, the function is going to end up changing. And so when the secondary, tertiary, and quaternary structures are breaking down, when they're excessively heated, then the structure is gone, meaning that the function is basically gone, which is why the egg is going to end up changing, it's going to end up changing the color. Anybody else wants to take another shot on that? I don't know if I clearly explained it, so, you know.
I asked Miss Rao about it, and one thing she said, like, just to be really sure, is you could mention the specific, like, the types of bonds, like the, you know, the hydrogen bonds and the different types of bonds in the secondary and tertiary structures, you know, just to be safe. One thing she gave, piece of advice she gave me was just, like, bombard it with vocabulary that's really pertinent. That'll help you get a better score.
I'm not sure if this answer would work, but I know during class today she said that like high temperature like destroys like the structure. So I mean, obviously that's a bit vague, but like if you keep like at least that basic idea in mind, like you might get like a few points on this question. So that's just, yeah, that's just a simple way of, I mean, thinking about it. Obviously, I think it might be better just to like know about like all the different kinds of structures and like MG like said the bonds, but yeah, that's just something to keep in mind.
Okay, so for the next one, it says that the following molecules is sucrose, and we're talking about, so sucrose is a polysaccharide, and it's basically saying how is this created from monosaccharides. So it's kind of what we discussed before with the two, like what we were discussing with the glycosidic linkages. And so here we have, so this is a sucrose, and so we have, okay, can somebody actually, how do we tell if this is a dehydration synthesis or a hydrolysis, just like by looking at it? I'm not completely sure.
I think it's because because the question is asking how is the sucrose being formed from mono. Yeah, I think that's why it would be
dehydration synthesis. Okay. So what if we didn't get that? Basically, since it's saying created, we know that if it says created, we're looking at a dehydration synthesis because synthesis is creation. But with a hydrolysis reaction, we're basically breaking something, we're not creating something. So because it says that, we know we're looking at a dehydration synthesis, and then we can just explain that basically what happens is we have these two monomers, and the monomers are going to be joined together by removing an H2O. Oh, okay, so a disaccharide instead of a polysaccharide, because it's just two pieces.
And she also said that it would be good if you could name the two monosaccharides specifically.
So this is glucose, because it's 6. And then what is it? Glucose plus fructose, I think.
Yes.
Yeah, glucose and fructose. And I think on the PowerPoint there's a slide that like
what are the other two? There's
glucose
more.
Plus galactose is lactose.
Yeah.
And then glucose plus glucose. I'm not completely sure. I forgot what it is.
Yeah, I think there's one more.
There's.
Here. Glucose plus glucose is maltose. That's what it is.
Maltose. That's right. Okay. Cool. Thanks. Oh, there's one more thing. She said that once again, if you want to like go for just be extra sure, you could talk about how enzymes help in the process. Like in a dehydration synthesis, you need an enzyme to help it happen.
I have a question. Hello?
Yes.
Oh,
yes,
sorry. Okay, my
mic.
mic goes crazy sometimes, so I have to make sure you guys can hear me. So for the FRQ, does she say that there's like a certain way you have to answer it, or like, do we just answer it? Like, is there like a certain format or something?
I don't think there's any kind of format as long as you like answer the question. Like it's kind of like like the DBQ from last year, like as long as you have like all the correct parts, like I don't think you have to like write a whole like introduction paragraph and like have like apes and aces and stuff like that, as long as you have the right answer. Because like that's what science is, right? Because like you don't need, it's just about being concise. So yeah.
Do we need to know specific molecules for the test? Like, do we need to be able to identify glucose and fructose?
I think she said we need to know glucose because it's a 6 carbon, so looking at like the hexagon or like the C6. And then I think she said that we should know those ones that we were just talking about, like glucose plus glucose. I'm not sure. I think it's, I remember saying something, but I'm not entirely sure. Did I freeze?
Yeah, you were breaking up a lot.
Yeah, you did.
Well, I was saying, I think we need to know glucose, and then I think it's important to know like the glucose plus glucose yields maltose, like we were just talking about. I'm pretty sure she said we should know that. So those ones on that slide. So,
Thank you.
yes. Okay, so let's do the next one. So it's how are cows able to digest cellulose? And so I think we should go to the PowerPoint because that is very well explained there. So we talked about how, so I think we should go here actually. So so carbohydrates and the polysaccharides are the polymers, And so there's basically 2 different roles. They can be storage or structure. And so when we're looking at the storage and structure, we have starch and glycogen. And so starch is your plant storage and glycogen is your animal storage. And so starch, and correct me if I'm wrong, but starch is part of the, like, it has an alpha glucose, if I'm correct. Yes, and then we're looking at something like cellulose. It actually has, is this the beta symbol, or am I being crazy? I think so. So that's going to have, we don't need to know that, but it's basically, it's like a straight structure. And because of that, what happens is that we're not able, the enzymes that are digesting the starches, because it has that alpha linkage, we can't hydrolyze these linkages that we have here. And so because that, we're not able to simply like eat it or things like that. So cellulose in our human bodies, we're able to eat it because we have fibers, or it goes through our body as a fiber. But in animals, we actually don't have that. So cows are herbivores, and you often see cows eating grass. So how does that happen? It's because they basically have a symbiotic relationship with microbes. And so these microbes are in their bodies and allows them to digest things like cellulose, which is why you have a lot of herbivores, because you know, you really can't just eat straight grass. The reason why they're able to do that is because of those enzymes. That answers 3. How are they able to digest cellulose? So how are they able to digest plants?
I'm gonna copy and paste an image that I searched up real quick. I think what you said was right about starch having the alpha glucose monomers, and then cellulose has the beta glucose.
Okay, yeah. And so these ones, the alpha ones, we're able to digest, whereas these ones that we're not able to unless we have the, like through insoluble fiber or through the microbes.
Could you talk about the structure of, what was it? So we talked about starch, cellulose. The other ones are glycogen and chitin, whatever you call it.
So that's like, it's kind of like 2 sides of it. You have like starch and glycogen. No, yeah, starch and glycogen. Then you have cellulose and chitin. We can talk about that one.
Okay.
So yeah, this is an example that cow, it's because they have the microbes that they're able to eat it. And so yeah, I don't know how to say it. I'm going to say this chitin. I don't know if that's correct, but it's another structural polysaccharide, and we see it on the exoskeleton anthropods. In one way that, maybe, I don't know if she's going to ask this, but if you want to distinguish it, you know that it has a nitrogen. So if you see the nitrogen or you see the N, then you know it's going to be chitin or kitten. And that's what you see in the cell wall of fungi. So cellulose is in the cell wall of plants, and chitin, kitten, is in the cell wall of fungi. So yeah, so this is kind of how it looks, and so you can see how it has the nitrogen, so you have that, and you can see how it's part of the exoskeleton of anthropods, and it's used to make thread.
Is chitin the one? There's one that goes like, I know there's one that goes like alternating. Is that chitin or glycogen?
Well, what just happened? I'm so confused. Okay, I'll go back. I'm not sure. Like, what do you mean by alternating?
Like, in the, you know, the monomers, like, their orientation alternates, I think, on one of them. What chapter is this?
It's chapter 4 or chapter 5?
It's chapter 5, yeah. I think it might be in the PowerPoint.
Well, while we were figuring that out, let's continue. Okay. Explain briefly how normal red blood cells in our hemoglobin can turn into sickle cell red cells. So if you were in the, I think we did this in the, like in bio today, we discussed this concept. And so again, the PowerPoint does have a really good answer to it, so you can just look at that. So basically what we talked about was in proteins, you have your primary structure, which is just like your sequence of amino acids. You have your secondary structure, which is like the folding and the folding. The tertiary structure is going to be the, I always forget this, it's the interaction among the R groups. So just remember that, oh, you can't see the PowerPoint? That's so sad. Okay, let me see. Share screen. Can you see it now?
Yeah, we can see it.
Oh, okay. So like I was saying, the primary structure is just your sequence of amino acids. The secondary structure are your coils and folds. Your tertiary structure is your interactions among your R groups. So remember, in your protein, you have a carbon, you have a hydrogen, you have an amino group, or an amino, and then you have a carboxyl. Is that correct? Yeah, a carboxyl, and then you have an R group. And so the R groups are what vary and what creates the sort of differences in your amino acids. And so the interactions among the R groups is your tertiary structure, and your quaternary structure is just multiple peptide change. So if you look at the cell and the sickle cell here, you have the primary structure where you have these, but you can see how in the, you don't need to know this specifically, like what the structure is, but in the primary structure, we have a GLU and it's being exchanged with the VAL, and that's going to create a really big change and cause sickle cell disease. And so you can see how the shape is going to change, and yes. And so in a regular cell, the molecules are able to associate with another and carry oxygens. When you have sickle cell disease or sickle cell anemia, basically what happens is that these molecules they crystallize, and so they're not able to carry the oxygen correctly, and because of that, the shape of your cell is going to change.
It was cellulose, the one that was alternating. So beta glucose, when it forms that glycosidic linkage, it goes alternating. So like when you go back to that image, you'll see like the oxygens, the oxygen in the 6-carbon ring. You'll see the oxygen at the top first, then at the bottom, then at the top, then at the and so on.
I have a question. So can you re-explain what it means by like crystallize? Because I don't really get what that means.
Okay, so in this one, it says that, so in the normal hemoglobin, the molecules do not, molecules do not associate with one another. So each molecule kind of stays in its own little sphere, and they each carry oxygen. But when you look at it in the sickle cell, the molecules actually do interact with another, and what happens is they crystallize into a fiber. Not entirely sure what that means, but when they crystallize into a fiber, what it means is that the capacity to carry oxygen is reduced. So before, each one was carrying its own specific oxygen. When they interact, we're not able to carry as much oxygen. And I don't know if anybody else wants to add on to that, because I didn't fully.
Yeah, like I think MG put it well. He said like they clump together. I mean, yeah, so Mr. I don't know if Mr. Rock explained this, but like I think what it means by crystallize is like, yeah, it clumps together and it kind of like hinders the ability to like carry oxygen. Like each one of them should be able to carry, like you said, Anna, carry like oxygen on its own. And if it's all stuck together, that sort of like doesn't allow the oxygen for them to carry oxygen because it's all stuck together. So yeah, I think that's sort of what that means.
Kind of like if you have like a ball pit, or if you have like a jar and there's one ball in it, it's easy to like pick it up. But if you have so many different things, it's a lot harder to pick it up. So that helps.
Yeah, I think I get it now.
Okay, so just a quick note before we move on. If anyone has any questions or things they want to focus on, we can just move over to that because the FRQ isn't as big as a priority as like working on the actual topics are. Anna, would you mind like moving back to the FRQs?
Not on there.
No, we're on the PowerPoint still.
So fun. My screen sharing is very weird. Okay. Is it there now?
Yeah, okay. So explain briefly the tertiary structure of proteins. Okay, so I wasn't really sure about this one, but I think it might explain it on the PowerPoint. But which chapter is? I think this is chapter 4, right? Chapter 5? Okay. So which slide is this? Let me see.
Do you see it now? The slide.
Yeah, I can see it.
Okay.
Okay, so this is a tertiary slide. Okay, so if anyone wants to jump in, you can, because I'm honestly not really sure about this one. But they ask why is it stable? So I think it might have to do with so they say the interactions among R groups. So okay. So okay, I'll be completely honest here. I wasn't really sure how to answer this question. I said before, but I mean, okay, here it is.
I kind of have an idea, so I can like jump in. I think today in class she said that there's the Disulfide Bridge, and so you kind of have like a bunch of different things and like folds in the secondary structure, and what really helps and holds it all together is the Disulfide Bridge. So that's really stable.
And she said since it's a covalent bond, like it's much stronger than the weak bonds in the van der Waals and then the hydrophobic interactions and the hydrogen.
So are all of these here covalent bonds and then these dotted ones hydrogen bonds?
So it's the ones that are dotted are always hydrogen, and those are always intermolecular, not intramolecular. The hydrophobic interactions just happen from the stuff we were talking about last year in chemistry, which is like the London dispersion forces. You know how there's the temporary electron cloud polarizability, so that's how that happens. The reason I got confused today in class was because I would think that the ionic bonds are stronger because in chemistry how we learned that NaCl that bond is stronger than a covalent bond. But in this case, what she told me was that it's kind of like the hydrophobic interactions where the side chains are just kind of interacting they're kind of mingling with each other. They're not fully bonding, but it's still an ionic bond because they're charged, if that makes sense. So that's why I think she said the disulfide is stronger, because it's covalent.
So I think that answered it pretty well. Explain the brief tertiary structure of proteins and why is it stable. So this is the structure, kind of you have like different hydrogen bonds, you have different interactions, and then the reason why it's stable is because you have the disulfide bridge.
And she said the disulfide bridge was like a combination of the thiol groups, right? Like
Yes.
sulfide, yeah.
Because so SS and H is the.
Wait, did she say anything about? Did she say anything about the hydrogen bonds when she was explaining this? Like I know you said that she said it was based on the covalent bonds that was stable or whatever, but I don't know if she said anything about the hydrogen bonds.
Do you want me, sorry, are you addressing that
I
to
don't
me? Or?
remember.
I'm asking anyone.
Oh, okay. And I think you're frozen. I don't know if it's just me again.
Yeah.
Am I frozen?
Yeah, you're frozen, Anna.
Can you hear me now? No?
Yes.
Yeah.
Can you hear me now? Oh,
Yes, we can
well,
hear you
I
now.
wasn't saying anything, so. Okay, well, I wasn't saying anything relevant, so.
Okay.
I think she just said the hydrogen bonds are there, but they're not as important as because the hydrogen bonds are like intermolecular versus covalent bonds, which are intra, meaning like they literally form an actual bond that creates a molecule.
I think the most important thing is just knowing that there's the bridge. And just knowing that it's
Okay, yeah, that makes sense.
We can speed through this one. So describe why water is an ideal medium for living things. So just the 5 properties of water, and I think we'll talk about those, and it's in PowerPoint 3. There's those things, and then describe 3 types of chemical bonds interactions found in proteins. So I think for this one you could talk about the R group, right? And you can talk about the different interactions, are like the van der Waals interactions. On the PowerPoint, it specifically says R group. That's what the R group does.
We can actually open up. Can you guys open up that link, because it's directly from College Board's rubric, and that. I think that'll help. Huh?
There's answers.
Yeah, it's just for that one though, not for all of them.
Oh, okay. Okay, yeah. So this basically what was in that PowerPoint. So the hydrogen bonds, the bridge. So basically anything that you see in that structure picture that you said, we can talk about, as well as the R group. So yeah. Oh.
Can we go over the last slide on the PowerPoint with the chart?
Yeah, let's go over that. Okay, basically what it's saying is that this is the big very broad term are the carbohydrates, lipids, proteins, and nucleic acids. And all of these are macromolecules, but technically a lipid is not a polymer because it's not made up of repeating small units. And so the monomers are, again, the small units, and so you have monosaccharides. And monosaccharides are, if you want to think about it based on the functional groups, this has to do with the carbonyl group, because the carbonyl is with the sugars. The fatty acid is going to be made up of a glycerol, so a hydroxyl group, as well as a carboxyl group. No. It's not made of a glycerol. It's made of a carboxyl group as well as a hydrocarbon chain. Is that correct?
Are you talking about the fatty acid chains?
Yes.
Like the triglyceride, or just the
fatty acid.
The fatty acid, I I might be wrong. I think it's just the hydrocarbon chain, right?
Doesn't it have a carboxyl with it?
You might be right. I think it. You're right. You're right. I think it does have the carb because the carboxyl would be then what joins with the glycerol, right?
Yeah.
I think so. Okay.
So monosaccharides is made from the carbonyl group, then several of them join together and form polysaccharides with glycosidic linkages, like we talked about. Then the fatty acid, and then when you add in the glycerols, when you add in the hydroxyl group, it becomes a triglyceride, and that's ester linkages. We didn't really talk about those, but I guess just know that it's an ester linkage. And then amino acids are going to be your carboxyl group, so the carboxylic acid, and then your amine group, which is your NH2, which is like your base. So it's a weak base and a weak acid, so that's an amino acid. And then several of those join together to form polypeptides through peptide bonds. And then nucleotides are your monomers here, and then several of them joined together are polynucleotides, and then this is the linkage. And so carbohydrates are a polymer, proteins are a polymer, and nucleic acids are a polymer. And then you can I was confused about this, you can say like either or, but these are more like the broad term. So yeah.
Wait, what are ester linkages?
It's like what links the different monomers together. So what links the different fatty acids to form the triglyceride. I think there's an image. We didn't really talk about it, so I don't know if it's like very relevant.
So I'm not sure, but I think, I mean, I might be wrong, but I think it's the, you know how you're talking about the carboxyl group?
Mm-hmm.
Yeah, like right there, how the carboxyl group bonds to the glycerol in the top. So that glycerol in the top, in order for it to, like, do you see how it's CO? I'm gonna try and annotate these, and then I'll fix it. But like right here, like right this oxygen right here, originally it was a COOH, and then similarly this carbon here also had the COOH, I think, and or not OH. Maybe it was just an O, and then the HOH. Oh, it must have been an H, yeah, because that would have been the So they would have been the HOH, and then the HOH went away, and that was the dehydration synthesis. So that's an ester linkage, I guess.
It's a picture of an ester. I think.
Oh my.
I'm not sure what slide it's on, but I know like there was a label on a picture somewhere that showed it.
This is proteins. It should be somewhere here.
It's on slide 23.
Oh, okay. Oh, okay. So
Okay.
That's ester linkage. So this is the carbonyl group. Why is there a carbonyl group here?
It was originally carboxyl, right? So
okay, yeah.
now because of the dehydration synthesis.
So we took away so it was COOH, so we took away the H as well as the two O's. Okay. So then we form the ester linkage.
So that's triacylglycerol. So glycerol itself is just the thing in the left, right? That rectangle in the left.
Glycerol is an OH. So, but then this is after the
that's hydroxyl, right? I'm saying like the glycerol, like the whole thing is glycerol is that whole carbon chain on the, okay.
Yeah.
So does glycerol always have like 3 carbons, or can it have like more? Like instead of a triglyceride, could you potentially have like I have no idea if that exists, but like a pentaglyceride or something, and then you have like a glycerol on the side? Is that possible?
I have no idea.
Okay.
Wait, so this is a glycerol attached. This is just the glycerol.
Oh, okay.
Right? Is it? I'm not sure.
Yeah. That's just the glycerol. Yeah.
And then this here is the fatty acid, and then this is the carboxyl. It's joining, right? This is the carboxyl.
Yeah.
Synthesis.
So glycerol, on each of the C's, it has a hydroxyl. That's why it's glycerol, because it has the
hydroxyl.
I see. Okay.
That's what I think.
Yeah. And there's like different types of fatty acids, but I don't think we need to know them, right?
Just saturated, unsaturated.
No, I'm saying like there's like, depending on the length of the chains and whether it's double bonded or single bonded, like that one on the slide itself, I think it said palmitic acid or something, but like there's different names for different fatty acids, but I don't think we need to know them, right?
Yeah.
Cool. So essentially, they're all from dehydration synthesis. It's just called something different depending on the thing.
Yeah.
Okay, cool. The thing. Yeah. I couldn't find that one.
Okay, so we have less than a minute, so I guess we're gonna end. But if you guys have any questions, Ms. Rao is doing a session tomorrow, so you can go ahead and ask her that or email her. And yeah, so thanks for coming today, guys.
I think Shandana's gonna post this on the YouTube.
Yeah, I'm gonna post I'm recording. I'm gonna post this on YouTube, and don't forget to fill out like the Rose Now Google Forms, because attending these sessions will count as volunteer hours or CAS hours. So.
And subscribe to the YouTube channel.
Yeah, subscribe to the YouTube channel.